everyone. Welcome to Hooked on Ocean Acidification mini-series. I'm Kirsten Wakefield, Stakeholder Outreach Coordinator Liaison for MACAN and MACAN, and I'll be your host for tonight. We're thrilled to see over 150 people have registered for this meeting tonight. And while this workshop was designed with the recreational fishing community in mind, we've attracted a very diverse group ranging from social scientists to fisheries managers to oyster gardeners, educators, and watershed ambassadors. We're looking forward to a rich discussion this evening with everyone. Hooked on Ocean Acidification is sponsored by the Mid-Atlantic Coastal Acidification Network, MACAN, and the Mid-Atlantic Regional Association for Coastal Ocean Observing Systems, MACAN, in partnership with Rutgers Cooperative Extension, New Jersey Sea Grant Consortium, the Virginia Institute of Marine Science, and the Mid-Atlantic Regional Council on the Ocean. And I'd also like to thank our planning team members, Susanna Music, Mike Danko, Doug Zemeckis, and Kat Davis for their excellent help in organizing this program. If you aren't that familiar with MACAN, it's an association of scientists, tribal, federal, and state agency representatives, resource managers, and industry partners who seek to coordinate and guide regional observing, research, and modeling of ocean and coastal acidification. MACAN is co-coordinated by MACAN and the Mid-Atlantic Regional Council on the Ocean, and its efforts are focused on the waters south of Long Island to the southern shores of Virginia. The goals of MACAN are to develop a better understanding of the processes associated with estuarine, coastal, and ocean acidification, to predict the consequences for commercially and recreationally important fish and shellfish and other marine resources, and to devise local adaptation strategies that enable communities and industries to better prepare for changing conditions. In 2019, MACAN launched a stakeholder survey to try to gain a better understanding of the information needs in our region. Some of you may have even participated in that survey. And Grace will talk more about the results in her presentation tonight, but just to give a quick overview, we received a lot of questions from recreational fishermen about the science behind OA, the impacts it may have on recreationally important finfish and shellfish, and in what time frame we could expect to see those changes. There was also concern about whether acidification impacts would be more pronounced in coastal habitats versus open ocean waters. And several fishermen asked, how do our activities affect the acidity and what can we do about that? So in response to those questions, we've invited scientists at the forefront of acidification research in the Mid-Atlantic to discuss what ocean acidification means and what the risks are, to help us examine the vulnerability of coastal fishes to acidifying waters, to talk about how OA impacts shellfish and whether seagrasses can provide a local solution, and to demonstrate how 3D modeling can be used to forecast short and long-term trends in water quality and acidification in the Chesapeake Bay. Through the 4 evening sessions in Hooked on OA, we hope you'll take away a better understanding about how ocean acidification may affect the fish you target and what can be done to reduce the impacts. Before we get started, just a few navigational aids for the evening. We will be recording video, audio, and questions posted in the chat box during this presentation. So if you're not comfortable with that, please turn off your chat and video functions before we continue recording. Each presentation will be recorded tonight's especially and shared with participants at the conclusion of the mini-series. And that will be available on the MACAN webpage, and we'll provide that link at the end of the talk tonight. We have over 150 people registered for tonight's session, so we ask that you please mute yourselves during the presentation, and so we don't have any dogs barking or dinner dishes clanging in the background tonight. If you've logged in with his email address to Zoom tonight, or with a nickname, please change it to your first and last name to help us with the question and answer and the raffle at the end of tonight's program. If you're calling in by phone, you can send a private chat message to our moderator, Mike Danko, to provide your name for the raffle as well. If you have questions during the presentation, and we hope you'll have a lot, please type your message to the moderator, Mike Danko, in the chat box. And we'll take a break halfway through the presentation from Grace, and again at the end to answer your questions. So with the focus of this workshop being on recreational fishing impacts of OA, we'll prioritize questions that are relevant to that topic. However, we will do our best to follow up on all of your questions. So if you don't get an answer tonight, please email us after the presentation and we'll follow up with you directly. We'll wind up the evening with a raffle for a $25 gift card to Bass Pro Shops. So stay close to your computer and don't wander away for a snack in the middle. And now I'm pleased to introduce Dr. Grace Saba. Grace is an assistant professor in the Center for Ocean Observing Leadership in the Department of Marine and Coastal Sciences at Rutgers University. Grace's research interests are in the fields of marine organismal ecology and physiology, with an emphasis on how organisms interact with their environment, how physiological processes impact biogeochemistry, and how climate change, such as ocean acidification and warming, impacts these processes. She integrates laboratory experiments, fieldwork, and ocean observation in her research. And her recent efforts result in the development of a novel pH sensor for autonomous underwater gliders, which she now uses to monitor pH changes in the ocean. In addition to her research activities, Grace co-founded the MACAN Mid-Atlantic Coastal Acidification Network in 2016 and continues to serve as both a member of their steering committee and science working group. She's currently advising New Jersey's DEP's coastal management program on incorporating acidification as an element of state coastal climate resilience planning. So with that, we'll go ahead and turn the program over to Grace.
Thanks so much, Kirsten, for that introduction. Let me share my screen here. Okay, can everyone see that? Excellent. Okay. Hi, everyone. Thanks for coming. I'll talk a little bit about the stakeholder surveys before I get into discussing ocean acidification. So with that, I'm just going to go ahead and start if I can. There we go. Okay, so Kirsten did mention that MACAN, in efforts to address gaps of knowledge for MACAN itself, because we weren't sure how to engage with stakeholders from the beginning. So what we wanted to do is do these surveys with a range of different stakeholders. So we had aquaculture, recreational fishermen, as you know, commercial shell fishermen, commercial fin fishermen, charter boat operators in the seafood industry. And we sent out several surveys and we got quite a lot of response. And we are excited to see that the recreational fishing community we got the highest responses from. We got 96 responses to those surveys. So the goal of the surveys, as Kirsten mentioned, was to better understand, what information or what knowledge these different groups had about ocean acidification, what were their concerns, and things that they would like to know about it. And that's really why we're here. I just put this map on here too to see just the range of places where these different communities were accessing, the fishing and the shellfish. So you can get, pretty much through the Mid-Atlantic region, all these different groups are occupying where they're fishing. Oops, why can't I advance with the arrow? Okay, I'll use that. Okay, and so this is, I just wanted to show a little bit of the responses from the surveys. So this was the types of fish or shellfish that recreational fishermen like to fish for. And these were the highest number that we got from the responses. So striped bass, flounder, bluefish, weakfish, black sea bass. And you'll see kind of going through, I wanted to just put this up because, we know we've had experiments done on some of these species, but a lot of these species we don't have information for and how they're responding to ocean acidification. So I wanted to just put that up to preface what I'm going to talk about a little bit later. And then getting back to how much these different fishing groups know about acidification to give us an idea of, what work we need to do in outreach and education is. So these are the results of which statement best describes your understanding of coastal and/or ocean acidification. And again, these are the different different groups that we surveyed. The aquaculture industry had the most knowledge for ocean acidification, and then recreational fishermen had the least knowledge. So now it's good that we're targeting this group so we can provide you with some of that information and help you understand what it is and why we're concerned about it. So I just put this slide back up again. This is the slide that Kirsten showed with the questions that she already discussed with you. So I'm hoping that my talk today will answer some of these questions for you. There's still a lot of things we don't understand, so I can't answer all of them with great detail, but I will do my best. So I will give kind of an introduction on what ocean acidification is, what we know about as far as the trends of ocean acidification, how coastal acidification can impact on smaller spatial scales. We'll talk today also about how ocean acidification impacts organisms, and then we'll go on from there about what kind of the steps we're taking now to help with the ocean acidification problem. So the Earth has a natural greenhouse effect, which in this picture is in sort of an equilibrium. So carbon dioxide, or CO2, is taken up by plants and phytoplankton through the process of photosynthesis. And then the CO2 is respired by organisms. And the ocean exchanges carbon dioxide with the atmosphere, and the ocean plays a really important role in the global carbon cycle because of that exchange with carbon dioxide with the atmosphere. So CO2 it's a greenhouse gas, which means that it traps heat in the atmosphere. So it actually makes our planet habitable to life as we know it. But too much of it is not a great thing. So what happens is, and what's been happening since the end of the Industrial Revolution in the 1830s, is that we've released a lot of this locked away carbon through things like fossil fuel usage and combustion, deforestation, cement production has released a lot of that excess carbon dioxide into the atmosphere, and it's being trapped into the atmosphere. And that's what's really causing warming globally. And so that's the first problem with excess CO2 in the atmosphere. The other problem is that because the oceans are absorbing that carbon dioxide, once carbon dioxide interacts with seawater, it can become more acidic. And I'll talk about the chemistry of that in just a little bit. So the ocean acidification is, we call that kind of the other CO2 problem, because we have warming in the atmosphere, but then we have this ocean acidification that can occur in the ocean. And it's really driven by that increased atmospheric CO2 that's coming in. And so the oceans are absorbing about 1/3 of the excess or human-caused CO2 in the atmosphere. And it's happening at a pretty rapid rate. And that's really the concern. So let's talk a little bit about chemistry. And it's, I think every audience needs to kind of understand a little bit about the chemistry, because some of the units that I'll be using and showing, you'll need to kind of understand how the chemistry works to be able to understand what those units are. And so what happens is when CO2 interacts with the ocean, the seawater combines with carbon dioxide to form a carbonic acid. So that's this kind of this equation here. So CO2 plus the seawater forms this carbonic acid. And then what happens is once it forms that acid, it kind of dissociates into these different species of carbon. So we get bicarbonate and carbonate ions. So these are these speciation here. And that releases these hydrogen ions. And that's how we measure pH. pH is a measure of, it's an inverse measurement of those hydrogen ions. So if you have more hydrogen ions in the water, it decreases pH in the water, which is a measure of acidity. So when we have this excess CO2 coming in, you're releasing more hydrogen ions, which decreases the pH and increases ocean acidity. So what happens though is that the ocean actually has a natural buffering process. And so most of that speciation of the carbon is in this bicarbonate form. And so what happens is, the ocean wants to have that bicarbonate. And so the carbonate will form with these hydrogen ions. And so you'll start losing these carbonate ions. And that's really important because if we have less carbonate ions, we'll have less for organisms to use. And what I mean by that is that a lot of these several marine organisms that form shells, things like our molluscan shellfish, corals. This is a pteropod, which is a type of zooplankton in the ocean that's really common. has a calcium carbonate shell. But so this carbonate binds with calcium to form calcium carbonate, this hard structure. And we call that process calcification. And so if there's less of that carbonate ion, it's harder for them to build those structures. And so we have what we call the saturation state, which is represented by this unit or symbol called omega. And it's if you have a lot of carbonate ions in the ocean, then we call it supersaturated. And so this omega value is greater than 1. And that means that there's no problems with finding that carbonate in order to build calcium carbonate shells. But if there's not enough of those carbonate ions in the water to form with calcium, then we call that undersaturated. So the water is undersaturated with respect to these carbonate ions. And so that's an omega value of less than 1. I will preface this though by saying that some organisms can be affected by omega values even around 3. So it doesn't have to be less than 1. So I think that's a really important note. You may not see, mortality, but you might see some subtle effects like they have issues with reproduction or growth rates or something like that. But this is these saturation state values it's a very common unit that we use in ocean acidification research to express really what is the impact of ocean acidification, because it's not all about pH, and you need several different measurements. So if you have pH, you can measure CO2, and the combination of those with some other variables like temperature and salinity helps you calculate saturation state, because you can't measure saturation state directly. So, okay, congratulations, we've passed the chemistry. So I just wanted to give you that, because I'll talk about pH, I'll talk about CO2, and I'm going to talk about the saturation state as well. So with ocean acidification, just to reiterate, we have more CO2 in the ocean, we have less pH, so the pH is lower, and the saturation state is lower. So moving forward, that's what I'll be referring to. So how is it changing? So this is a map or a graph showing CO2 concentration, and this is in the atmosphere from about 1700. And this data is taken from ice cores, because we didn't have sensors that we could put out way back then that measured atmospheric CO2. But we can get a record of CO2 that's trapped in ice. And so we can have this record that's showing changes in CO2 over time. Once we get here, so starting about 1958, we have a station out at Mauna Loa, it's in Hawaii, that's been measuring atmospheric carbon dioxide since 1958. So this is kind of the station that we use to look at changes. There's obviously CO2 being measured in a lot of places, but this is the one we've been using because it has the longest time series. But what you'll see is this pretty sharp increase in atmospheric carbon dioxide since about that time. And that's linked to just the increased usage of fossil fuels and deforestation and all the other things I mentioned before. So it's this rapid rate of increase is really what the concern is. And so I just put this box around here because the next figure I'm going to show is covering that time period. And so this is the red line here is that same atmospheric CO2 concentration increasing over time from 1958 to almost present. And then the green line is seawater PCO2. So that's what's the concentration of carbon dioxide in the seawater. And so you can see that's increasing at a pretty similar rate to the atmospheric CO2. So you can see that pair or the pairing or that synergy between the atmospheric and the ocean and how they're increasing at a similar rate. And then this blue line is pH. So now you can kind of see the relationships now between the increase of CO2 and then that's starting, you'll see that decline in pH over time. And so since the 1800s, since the end of the Industrial Revolution, or the beginning of it, sorry, not the end, atmospheric CO2 has increased about 40%. And that's equivalent to a drop in about a 0.1 pH unit drop in pH, but it's equivalent to about a 28% increase in ocean acidity since the 1800s. And that rate of change is about 10 times faster than anything experienced over the past 50 million years. So it's a very rapid rate of change. And this is occurring globally. So this is really what we term ocean acidification. So what are we expecting it to do? So we are expecting carbon dioxide or CO2 to double by the year 2100. And that will cause an additional drop in pH of about 0.2 to 0.3 units. So this figure is from the Intergovernmental Panel on Climate Change Fifth Assessment Report that came out in 2014. They're working on the next version now of that report. It happens, every so many years. And you can see the pH, so this is observed values, and then these are modeled values here. And these are a range of scenarios. So RCP 2.6 is something like if we stopped CO2 emissions now, and then pH would kind of level out over time. RCP 8.5 is kind of your worst case scenario. It's business as usual. So if we don't do anything to mitigate the release or the release of CO2 into the atmosphere, this is the trend line we're looking at. And so what we're expecting again is that drop in pH of about 0.2 to 0.3 units, and that's equivalent to about 100 to 150% increase in ocean acidity since the 1800s. So a very rapid rate of increase. So those are the trends, and those are what's happening globally. And so I kind of wanted to put this now into context of where we are, the Mid-Atlantic. And the Mid-Atlantic is situated here. And we have two kind of major source waters coming into our shelf. And so that is this Labrador Current that comes from the north. It's very cold. And I'll talk a little bit more about it. But then from the south, we have these southern waters that are highly influenced by the Gulf Stream. And so Mid-Atlantic here is really in this place where you're mixing these very different water sources. And so, we have to know a lot about not just what those water sources are like in terms of the chemistry, but we also need to know how they're mixing and how much they're mixing and how much they will be mixing in the future. So there's a lot of, it causes additional uncertainty because of this kind of unique combination of water sources here. So the Labrador Current that comes down from the north is cold. It has a lot more carbon dioxide in the water because it's colder, and colder water can hold more gas. And so inherently it can absorb more CO2. It's also weakly buffered, which means it's a little more fresh. And so what weakly buffered means is that it can't really buffer and help any changes in pH. So if it's impacted by CO2, the pH is going to change pretty quickly. As opposed to this warmer, saltier water coming from the south that's heavily influenced by the Gulf Stream. It's highly buffered. It has a pretty high pH. It has a high total alkalinity because the alkalinity of the water makes it more able to buffer out changes in pH. So, so you have these two different water masses that impact the Mid-Atlantic. And so the colors here are showing that omega value, that saturation state. And if you remember, less than 1 was something we didn't want to see. And greater than 1 is okay. But again, even values around 3. So in this kind of creamy color and this, well, I guess, bluish, I don't know, corn blue, I don't know what color that is. But here at 3, you can see that some organisms could still have some issues. So you can see the vast differences in that saturation state in these 2 different water sources that are mixing here in the mid-Atlantic. And I forgot to just mention that this is data from just one cruise that occurred in 2012. And these are carbon cruises that are now happening about once every 4 to 5 years. And we are seeing some more frequent cruises, so we can start looking at trends over time. Okay, so this is really where the Mid-Atlantic is going to see impacts of traditional ocean acidification, that absorption of CO2 into the water. But the Mid-Atlantic is also is exposed to a lot of other drivers that can impact ocean acidification. So and I'll kind of go through these. So this carbon dioxide here, so that's your traditional ocean acidification that is occurring globally. And the Mid-Atlantic has a lot of fresh water coming into the system, as you all know. And fresh water is naturally lower in pH, again, because it just doesn't have very good buffering capacity for dealing with pH changes. And so what happens is you get fresh water coming in, it's naturally lower in pH, and that's coming into our systems, our estuarine systems and our coastal shelf system. And then we have runoff coming out as well. And so that's coming out from our freshwater systems, and it has nutrients, which will promote phytoplankton growth. So this is this eutrophication here. And so these nutrients are taken up by phytoplankton, and they can grow into, pretty dense biomass in the surface water, which is good because they take up CO2. So they'll actually absorb CO2 in the surface, and pH will increase in the surface. But what happens is eventually they start to die off, and then they'll sink. And so once they start sinking, bacteria will eat them. We'll take all the particles and start breaking them down, and the bacteria produce a lot of CO2. And so you'll get kind of a very different CO2 from the surface to the bottom. And so in the bottom water, so the bacteria will take up all that organic matter that's coming down. They'll release the CO2, and it'll decrease pH in the bottom water. So we have to think about not just surface, but also the bottom. And then, of course, we have upwelling. It happens in the summertime. We get periodic episodes of upwelling along the coast. And that's when you go to the beach and you think it's going to be nice and warm because it's, July or August, and you jump in and it's freezing. And that's when the ocean is upwelling. So we have a very cold water mass sitting in the middle of the shelf at the bottom. It's called the cold pool. And what happens is you'll get sustained winds coming from the southwest, and it promotes this upwelling. And so that cold water will come up to the surface water. And that cold water is also inherently, because it's at the bottom, it's fed by these waters from the north, and there's a lot of respiration and CO2 and lower pH traditionally in that lower, colder water. And so that comes up to the near shore, and that can impact, episodic events of acidification near shore. So there's a lot of different coastal drivers going on in the mid-Atlantic. And so inherently, it's pretty hard to measure, but we are trying our best, and we're still trying to improve that. And I'll talk about that in just a little bit. So I'll talk a little bit about some observations recently that we've been able to take now with the underwater glider that Kirsten mentioned that's running in my laboratory. It's fitted with a pH sensor. So these are measurements that we make along the shelf waters of the Mid-Atlantic Bight. And then I'll talk a little bit about what's going on in some of our estuaries. So we can run some transects with these gliders. So gliders are, if you're not familiar with them, they're autonomous underwater vehicles, and they fly kind of in zigzag patterns up and down in the water column. They're buoyancy propelled. So that means that you can make the front end heavy or light, and just by pumping in some water and pumping it back out. And the wings on the side make it propel forward. So you get not only vertical resolution when you're sampling, but you also get spatial resolution. So what I'm showing you here are, we've done some transects across the shelf with the pH sensor. And so here are the depth profiles. So kind of just to orient you to the figure. So this is distance from shore along this axis. And so these are the shallow areas, and then these are the deep areas near the shelf break. And then this is depth coming along this axis. So you can see we get shallow and then we get deeper. And so we did these transects at 4 seasons, so we could see seasonal variability, which we didn't have before in this region. And we can kind of, gives us an idea of when should we be concerned, and how is that timed with organism life cycles and seasonal migrations of fish and things. So that's really why we wanted to look at seasonal dynamics. And so what we've seen and what, the lowest pH that we've seen and lowest aragonite saturation state that we've seen occurs during the summertime, which is expected because we have a lot of stratification on the shelf. And that just means because it's so warm in the summer, the surface water layer is very warm. It's 20 degrees or more at the surface. And so it kind of cuts off any kind of mixing from the surface to the bottom, because we have this big cold water mass at the bottom of the cold pool. And so there's very little mixing that happens during the summertime. It would take a huge storm to mix that water from the surface to the bottom. And so when we see that stratification, there's no ventilation, so the bottom water is not getting oxygen. And there's still all that biological activity happening at the bottom with the bacteria and the respiration. So they're releasing a lot of CO2. And so in the summertime, we see really low periods relative to the other seasons of pH and aragonite saturation state. And this is, where a lot of organisms live, specifically our molluscan and shellfish species. I circled just general habitat areas of our Atlantic surf clam and our Atlantic sea scallop, just so you can get an idea. And we care about this because we just want to see what conditions they're in. These organisms will spawn from spring through fall. So, we want to see what conditions they're in when they're spawning and reproducing. And then in the fall, we get big storms, as you guys know, and these storms start mixing in this water that's farther offshore. And if you remember, that's that Gulf Stream influenced water that's warm and salty and highly buffered and high in pH. And then that starts being mixed onto the shelf in the fall during these big storms that kind of finally overturn the water. So the surface and the bottom finally start mixing together. And that kind of alleviates the any kind of ocean acidification conditions that are happening on the shelf. So thank goodness for the fall overturn. So that's what's really happening on the shelf from the data that we have so far. We're still, we're using this data to understand kind of the baseline of what's going on right now, so that we can start monitoring over time to start seeing trends over time. And then, so that's the shelf area, and I'll talk a little bit about what's going on in some of our estuaries. So these are some studies led by Chris Gobler and Hannes Baumann, who will be giving a seminar. I think he's the next one. So he'll talk a lot about this with respect to impacts on fishes. So I just wanted to show you a little bit of the data so you can get an idea of some of the observations that we have. So this is a map showing some of the sites where they sampled, and I think the ones in this figure are just these kind of white circles with the black dot in the middle. And so the transects I'm showing are from west to east. So they're from here all, and then back to kind of the middle, going out to the middle of Long Island Sound. And so these are, so we have dissolved oxygen here on the left, if you guys can see my arrow, and pH is the right column, and then over time is from top to bottom. And again, these are depth profiles, so you can see from the surface to the bottom. And you can see that it's kind of a similar situation as in the Mid-Atlantic Bight, where it's the summertime where you start seeing issues with lower dissolved oxygen and lower pH, where you might be concerned about organisms living there at that time. And then, so I just wanted to show you that, and then also Chesapeake Bay. So this is a study done by Wei-Jun Tsai, who's at University of Delaware. He's done a lot of work in Chesapeake Bay and Delaware Bay as well. But just to give you an idea, so this is the Chesapeake Bay and its tributaries, and they did a cruise that just measured surface PCO2. So you can see these trends here. So you can see where the water is more fresh, at, coming in from all that fresh water coming down to the main stem of the Chesapeake Bay, where it gets a little bit more salty. So where it's really fresh is, again, it's that fresh water, it's lower in pH, there's less buffering capacity. So you see this high PCO2. There's a lot of organic material also coming in that can impact your carbonate chemistry. And I think these are two different time periods. I think this one was in August and this one was in April timeframe. But then looking vertically into the water column, here's, you can see dissolved oxygen here and pH. And these are just different dates. And again, this is depth coming down here. But you can see, it's higher at the surface and then it starts declining near the bottom. And here for pH, you see some kind of mid-water minimums for pH, and that's caused by just different bacterial activity that are mediating that kind of mid-water pH minimum there. So a lot of different things go into driving acidification. So there's the fresh water coming in, the biological activity, organic matter, upwelling. So there's still a lot of influences on acidification, and so it makes it difficult to measure and observe and really figure out what specifically is driving it. And it just turns out it's a very complex, chemistry problem, and a lot goes into it. So we're doing our best here. And so we have people doing the observations, and this is a map looking at observations in the Mid-Atlantic that MACAN worked with so this is data available on the MARCO data portal. It was a MACAN effort to kind of just figure out where are the observations for acidification in our region. And this would help us figure out what the gaps were. And so we did this exercise a few years ago, and we published a paper led by Katie Goldsmith. And it was really just, we wanted to know where things were being observed so we could start looking at what our gaps were to help start filling in those gaps. And so, the first glance, makes it look like we have pretty good coverage in the Mid-Atlantic. But some of these, like these green lines here on the shelf, a lot of those are from cruises that only happen, like I mentioned, once every 4 or 5 years. So we don't get a lot of good temporal variability. We don't understand really the temporal dynamics. We get the spatial, but not so much the temporal. We get a lot of measurements from moorings. These are things that are fixed stations, which give us, good temporal resolution, but don't give us the spatial resolution. And then we have issues where a lot of the measurements are from the surface, and we don't have a lot of measurements below the surface where, like I mentioned, the bottom can be problematic. So these are observation needs that we kind of concluded during that exercise that we're working towards filling. So high resolution depth profiling measurements, monitoring across the salinity gradient, because we have a lot of those in the mid-Atlantic with all of our freshwater inputs coming in. And observing ocean acidification with other stressors too is a big one, because ocean acidification isn't happening by itself. We're also seeing warming in the region. We see areas and time periods of low dissolved oxygen in our region. So there's a lot of things happening at the same time. And so, we also concluded that Mid-Atlantic would benefit from a comprehensive statewide or region-wide monitoring network that can cohesively act to address the observation needs. So that's something we're working towards. And I think I'm going to stop for questions if there are any. I will let the moderators tell me.
Hello, Grace. Just had a couple questions come in at the last minute.
Okay.
I'll just pull them up and. Sorry, I'm just having a problem. If you want to go on, Grace, I'm having some trouble. I saw them pop up and now they're.
Is that all right for me to ask it?
Yeah, go ahead if you have a question. Just want to identify.
To you, Mike. So, Grace, I teach high school chemistry in Japan, New Jersey, and I've done a lot of fishing though. But so what you're saying is, so due to the fact that the colder water dissolves, gases more regularly, that the CO2 remains can be produced more regularly at there, and that's why it's at the bottom, and then the production is happening at the seafloor, because I forgot what you said, but that's where it's happening. Does that make sense? Did that make any sense to you, Grace?
Yeah, now it makes sense, and it's very complex, right? So you just mentioned like 3 different drivers of carbonate chemistry in the water, right? So in general, colder water just absorbs more gas. It can hold and retain more gas, which CO2 is a gas.
Correct. Yes.
So if you have colder water, and especially coming from these northern regions like the Labrador Sea, Labrador Current, it's going to naturally just hold more CO2. The bottom water issue is it can be in the Mid-Atlantic. That is part of it, because we do have this cold pool in the bottom. It's not like that everywhere, but in the cold pool, it's 8 degrees in the summertime. So it's pretty cold, and it can inherently hold more CO2. But the biological activity is it's a totally kind of separate driver, right? So you have the production at the surface. The phytoplankton, again, they're taking up CO2 and they're releasing oxygen. So phytoplankton are very, phytoplankton provide about 50% of the oxygen we breathe. But they're also absorbing the equivalent of that in CO2 through the process of photosynthesis. So in the surface, you'll actually see the reduction of CO2, the increase in pH from that process. But then once that material starts sinking below the surface to the bottom, so that's when the bacteria take over, and the bacteria, so they take up oxygen and then they release CO2. So it's the opposite.
Right. So then they're in the bottom in the colder water, and then that colder water can actually hold more of the CO2, which creates the acidification when that CO2 breaks down.
Yep.
Thank you very much. I'm going to use this for my students, so I just let you know.
Oh, good.
Grace, we have one more question.
Okay.
Somebody, Grace Ann Taylor, had asked if the stormwater runoff also contributes additional CO2 and not just to the eutrophication to the water body.
Yeah, I mean, it can contain a lot of organic matter, which can contribute to the carbon, so the dissolved inorganic carbon and dissolved organic carbon. So it can contribute more of that into the system. I'm trying to think of a good example. I know, so the Pine Barrens in New Jersey, the water in the Pine Barrens is pretty, it's naturally acidic, like it can get really low, like below 6, a pH level below 6, which is pretty acidic. And that's because there's a lot of organic matter from all of that, the decomposition of all of those pines and the wetlands there that can contribute and cause that reduction in pH. I don't know if that helps.
Okay, great. So we have one more question just came in.
Mm-hmm.
It's from Carl Fredericks. He wants to know if climate change, increasing ocean water temperature, will counteract the ocean acidification because of less CO2 that can be held in the water.
Yeah, it's a good question. Hi, Carl. It's a good question. Yeah, I don't really know the answer to that, because the way I think of it is it can impact, I'm trying to think now, because it can impact the alkalinity of the water also once it's warming. And then the so the warming is happening at the surface of the ocean, but eventually it's being mixed and it's going to warm the full vertical water column. There's still going to be stratification, but it just might be less strong. So most of the warming that's going to happen is at the surface. So you're still going to see acidification at the bottom, but yeah, I really don't know the answer if that's going to counteract it completely. Yeah, because I always think of it in terms of the organisms, because it's warming at the same time that there's carbon dioxide in the ocean. The carbon dioxide is still going to be absorbed by the ocean, but maybe it just won't hold as much. But there's still more CO2 coming into the atmosphere, so it's got to go somewhere, which is the ocean.
Okay, Grace, that's all we have 2 other questions, but they're related to behavior change. I think you might be covering it in your next segment. So I'll hold those off until the end.
Okay, great. Okay, so I'll move on then. Okay, so we've talked, I've talked about kind of what we're seeing in the water, what the trends are, and now kind of getting into, why do we care? And really it's how it might impact organisms in the seawater. So this is looking at, so I've talked a little bit about the calcification process because I had to go into that when we're talking about the chemistry, but that's not the only process that could be, or that is impacted. So this is looking at, so this is work from Christy Croker, who did a kind of a synthesis of studies. So it was a meta-analysis that she did. So she incorporated all of these studies that looked at impacts of ocean acidification on organisms and the different processes that it might have impacted. And this, I just will explain this here. This is mean effect size. So it's basically a log response ratio, which means that anything below 0 is a negative impact. So you're looking at something that happened in the treatment, which is the ocean acidification, compared to the control. So anything negative or less than 0 here is a negative impact of ocean acidification. And she was looking at data and experiments that looked at different processes. So survival, calcification, which I talked about earlier, growth, photosynthesis, development, abundance, metabolism. So a whole range of different processes that are important to organisms. And you can see just visually from this graph that a lot of processes, a lot of studies found these processes were affected negatively by ocean acidification. There are a few exceptions to that. So photosynthesis is not too affected, and that's a lot of the reason why that is because phytoplankton actually, they absorb CO2, right? So in some studies, phytoplankton there was no response to that, and some of them actually had positive responses to ocean acidification because there was more CO2 for them to take up and grow. So there's, and you'll also notice the large range in these studies, in the responses between these studies. And, that's inherently been a challenge for ocean acidification research, because we've noticed that even within species, we see different responses. In between individuals, we see different responses. So there's such high variability in the responses that it's been a real challenge for ocean acidification research. So, okay, so we see these, but some of these aren't reported, some of the responses. So now we're seeing a lot of the studies are looking at impacts on reproduction, olfactory, so this kind of sense of smell, behavior, swimming ability, susceptibility to disease. And so these are all things, again, that we've seen some negative impacts of ocean acidification, some with no effect, right? Again, a very high range in responses even within these different processes. And so, we can do these in a lab. We can put an organism in a lab and measure these things under controlled conditions. But it's really hard to do these in the field. And so you want to do that so that you can really see if these are realistic responses. You want to see interactions. A lot of, these here, these are direct impacts, right? But we know that with other aspects of climate change research, with temperature and dissolved oxygen, there's indirect or subtle responses of these organisms that we may not pick up in a laboratory setting. And these are things like, biotic interactions. So if something has a lower growth or lower reproduction, if an organism has like issues with their sense of smell, will they be more susceptible to predators? And that's really hard to test that in a lab. And then kind of long-term and larger scale effects are, it's still questionable and still really hard to observe in the lab and the field. So questions about biodiversity. So how many species, how will ocean acidification impact the number of species in any given location? Ecosystem, so how is everything tied together, and we also don't know a whole lot about how organisms will acclimate or adapt to ocean acidification over time. Will they be able to do that at the rate of change we're seeing with ocean acidification? So that's still, these are some of the bigger questions we have still in the field. Ocean acidification really it's still in its infancy of research. It's only, some of the first studies were done specifically looking at ocean acidification only 30 years ago, or maybe a few years more than that. So it's a pretty young research level. So we're still learning a lot. So the general takeaways regarding acidification impacts on organisms, some of these are consistent and some of these aren't. So highly variable responses between species and even individuals. So this is challenging because it means that we can't assume that a similar species will respond the same. And that makes it really hard to generalize impacts on populations and communities and ecosystems. So it also, we bear the burden then of having to individually test these species in a lab, which is, really challenging. We need a lot of money for that. And that's, we don't always have a lot of money to do that type of research. So that's really challenging. But what this does tell us is that over time, there will be winners and there will be losers with respect to ocean acidification and climate change in general. Young life stages seem to be the most susceptible. Now, this is pretty consistent in all of the studies done so far. It's pretty consistent finding, and that's between several different types of organisms. But the young life stages just seem to be more susceptible. And that's a lot of that has to do with these larval stages don't have the energy reserves to be able to, and they need those energy reserves to grow to the point where they like start feeding or something, or grow like an oyster larvae where, they need those energy reserves the first few days of their life cycle to build their shell. And if they don't have that, if they don't have enough energy to do those things and deal with their environment that could be stressful, perhaps from ocean acidification, then they're going to have a really hard time. So that's pretty consistent. The effects are typically subtle and even indirect. So again, this is what I mentioned before, that an organism is not going to just die from ocean acidification. It can, but most often it doesn't. And it can have things like impacts on growth or reproduction, where maybe they don't produce as many eggs, or maybe the eggs don't hatch this as much under ocean acidification conditions as they would under non-acidified conditions. And so that will have, kind of longer term impacts on a population. So those are really hard, again, to detect in the lab. Ocean acidification interacts with other stressors. I mentioned this before, but yeah, it's not happening by itself. It's happening in coastal areas with low dissolved oxygen in bottom waters. It's happening with warming temperatures globally, but also regionally. We see, temperatures are definitely increasing in the mid-Atlantic bight over time. So how do those interact, and how do those maybe counteract? Like Carl was asking in his question that I couldn't answer very well. But these are still things that we're still trying to figure out. And we don't have a lot of research looking at those what we call multi-stressor impacts on organisms or the ecosystem itself. Food availability is very important to potential acclimation. This, again, is another very consistent thing that we're seeing. And that's because, again, like I mentioned before, you need those energy reserves to do what you need to do to survive, to thrive, but you also need those energy reserves to handle stressful conditions. And if you have enough food, then studies have shown that you can tolerate those conditions pretty well. If you don't tolerate them as well. So food availability is very important. So that's another aspect of research that people are looking into. Do we have enough food to support increased metabolic costs in the future? So what's happening in the mid-Atlantic to our species? So another MACAN effort was we did a kind of a synthesis of research on ocean acidification for mid-Atlantic species. And again, this was a few years ago. And so we compiled as much data as we could find regarding acidification or multi-stressor studies conducted on economically important groups and species in the mid-Atlantic. What we found was studies done on 18 different species, a range of different types of species from 59 studies. And most or all of these were species managed by the Fisheries Management Councils or NOAA, Northeast Fisheries Science Center, or states. And then these studies had a wide range of response variables. So it could have been anything from growth to reproduction to mortality. But we summarized those responses in this paper, and just showing a very simplified kind of finding of that was about 65% of those studies or those species had negative effects on them. 30% had no effect and 5% had positive effects. That 5% was phytoplankton and seagrasses. So all the photosynthetic organisms that take up CO2 had actually positive impacts. The rest were either negative or no effects. So we were able to synthesize that to at least kind of give us a broad view of, you know, is this okay, or is this not looking so great so far? And I'll talk a little bit about some of the specific things we saw for some of the recreational fish species, but I'm not going to go into too much detail because Hannes Baumann will probably present all of this at the next presentation. So, but I just wanted to give you a few studies that we looked at in that review paper. And one of them was the Chris Chambers' paper from 2014, where he was looking at the impacts of ocean acidification on summer flounder larvae. And what he found was that at low pH, there was decreased embryo survival. So that's really what's kind of shown in this figure. So you have CO2 level going from low to high, and then survival to hatching. So this would be 100% survival. And then this would be, what is it? Like almost 20% here. So he saw that decreased basically hatching success or embryo survival. And the larvae at hatching were larger, but they had less energy reserves. And again, this kind of goes back to, well, they probably had to utilize some of that energy to either deal with the acidification or grow larger. And then higher number and severity of tissue and organ malformations. Now, this is one that we've been commonly seeing in the shellfish is, you know, deformations of their shell structures. A lot of the calcifying organisms were seeing that. So that was one study that looked at larvae. But here's one that looked at summer flounder juveniles. And so this is looking at, so they looked at, this is a multi-stressor study, which is good. It was looking at both DO, dissolved oxygen, and pH impacts under different ranges of dissolved oxygen. So I guess what I really wanted to point out is that the mean, so this is showing mean specific growth rates, these numbers in the table. And they were always lower after longer exposure times because they wanted to see if, well, maybe over time they can acclimate to you know, tolerate these changes. But it didn't seem to be that way because at every period where there was a longer time period or a longer exposure time, the growth rates were lower. And they were lowest at the extreme dissolved oxygen and pH, but they were driven mainly by dissolved oxygen, not necessarily pH. Because if you go back to here, you can see that at extreme pH, growth rates are pretty similar, at least to the static or the ambient pH. So most of these changes in growth rates, these declines in growth rates were driven more by the dissolved oxygen, actually. But they did see high mortality at those extreme levels. But so these are juveniles, and we saw the larvae ones before. And then I'll just show one more fish species. So this is striped bass. So this paper by Linwood Hall was published in 1987. And they were really just trying to look at, because there were reductions, they were observed reductions in striped bass populations over a period of time. And they had linked them to larval mortality from acidified conditions. So they've had the data and they were trying to correlate those to larval mortality. And they did some lab experiments and saw some negative impacts as well. But then in a study done with juvenile striped bass, that even at the extreme low levels of dissolved oxygen and pH, they were fine. There was no effect on survival. So, you know, again, these are the same species, but different life stages, and you're seeing some kind of variability in their responses. And, you know, so this is, again, a challenge for this. So we have, you know, one species, but you're seeing different responses. So not only do we have to, you know, directly look at individual species, but we also have to think about their different life stages too. So that adds a whole level of complexity. So we have some significant research gaps though still, which I mentioned before. So this is a table that we put together for that review paper. And of the 35 managed species in our region, about 69% or 24 species had not been investigated for acidification impacts yet. And that includes, you know, some of these species that the recreational fishing community expressed, you know, that's what they're fishing for. So black sea bass, there's another one on there, the bluefish. I don't know if the weak fish was on there. I can't remember. Tautog was on there. So again, quite a hefty list of managed species that we have no idea how they're going to respond to ocean acidification. So this would be, you know, the list that we would kind of take forward and tackle this list first, if we had to in the lab. And then just some general research needs that we put together from that review. You know, we need the new studies focused on these species, but these need to include the various life stages, how well they can acclimate or adapt, potential thresholds of acidification. So what's their breaking point? At what point or what level of acidification is there the point of no return for them? And then these kind of interactions, how does that impact the food web, population dynamics, and community structure? You know, we really have not much information on those kinds of interactions. You know, we also want to look at mitigation strategies, specifically for aquaculture facilities, hatcheries, nurseries, and impacted waterways. And then we want to connect organism and ecosystem responses to ecosystem services and the economy. So how much of the impact on the organism might affect fisheries and the economy? So those are some pretty big questions. And we care because we have this ocean acidification and coastal acidification happening. We are seeing biological changes from the studies that we're doing. And then this will in turn have these socioeconomic impacts on our fishing community, our general public, people that eat seafood. And then, so now it's kind of spurred some of these actions, these management actions by states and individuals as well. And I'll talk a little bit about that. This paper came out in 2015 looking at social vulnerabilities from ocean acidification. And I just wanted to highlight that in the Mid-Atlantic, we were considered one of the highest social vulnerability locations, mainly because of the multiple drivers of ocean acidification in our region, but also our very valuable fisheries. So specifically the shellfish fisheries. So we are put in this pretty high social vulnerability category, which is significant for us because it, you know, really makes us, it really motivates us to do the research. There's a lot of text on the slide, but I did just want to list some commercially important shellfish species. And then you guys all probably know the commercially and recreational important finfish, and I've only listed just a few. So don't be offended if I didn't put your favorite one on there. But, you know, the marine resources in the Mid-Atlantic have these ecological significance, but also this economical significance. So this is just showing total landings revenue in the Mid-Atlantic from 2011 to 2015. You know, it's a lot. It's a lot. The Mid-Atlantic has brings in a lot of revenue from fisheries. New Jersey itself, the commercial fishing industry is the 5th largest in the US and provides more than 50,000 jobs. And then what's great about the research now and the direction that it's moving in is that these economic scenario analyses and vulnerability assessments, of kind of that figure that I showed you just before, they're starting to include acidification. So it should give us some idea of some potential socioeconomic impacts. So, you know, what are states doing? So I don't know if you guys are familiar with what happened in the Pacific Northwest a while back. So there's several shellfish hatcheries here in the Pacific Northwest, and they suffered an 80% decrease in oyster production in 2008, 2009, directly linked to ocean acidification. So they have a lot of that upwelling there. And what was happening is there was some really big upwelling events, and those upwelling events in the Pacific Northwest last a very long time. And their intake line to their hatchery was pulling in this water that was coming up from the bottom that was very undersaturated. So it had the low omega values. And they saw these shell deformities. They saw really high mortality coming, you know, in their hatchery. And so the industry actually took action, and they, you know, talked to their policymakers, and they really drove these policy actions in Washington. They formed this Ocean Acidification Blue Ribbon Panel, which convened in 2012. And then they developed from that the first state ocean acidification action plan. And then that kind of led to this region-wide impact it led to this multi-state Pacific Coast Collaborative that consists of Washington, Oregon, and California. And so they're really being proactive now about ocean acidification research. And then several other states in the US followed suit. So New York is developing their ocean acidification. They developed an ocean acidification task force that's now developing their ocean acidification action plan. Many other states are doing the exact same thing. And those that are developing the ocean acidification action plans have joined this International OA Alliance, which is basically like a promise to help reduce ocean acidification. There's been bipartisan support for ocean acidification related bills in the House committee. And then now, you know, we've developed these regional acidification networks, such as MACAN. So there's been a lot of efforts now to start just informing people about ocean acidification, talking to our policymakers, you know, what can we do on a state level, and now an international level with this OA Alliance. And NJDEP is recognizing the ocean acidification risks as well. So there was discussion of OA in their 2020 scientific report on climate change. It came out last year. And then in response to that, they developed the 2020 Global Warming Response Act 80/50 report. And as part of that, the Coastal Management Office is trying to incorporate ocean acidification into their plan to try to be proactive about climate change in New Jersey. So we're moving towards these goals. And on an individual level, I always get people ask me, like, what can I do? And I always like to end these talks this way, because, you know, it's not all doom and gloom. You know, things are happening and we are concerned about them, but we're taking action and we're doing what we can do to be proactive about it and see if we can help. And so even on an individual level, there are things that you can do, and I've listed them here. You know, using less fertilizers on your lawn, LED lights, mass transit, so driving less. So pretty straightforward things that you can do even on an individual level to help. So that's it. That's what I have. I hope I'm okay on time. Don't even know how much I went over.
No, you're fine, Grace, and
Okay.
thank you very much for your presentation. We appreciate it. We do have some questions. One is from Professor Judy Weiss. She had a question that some years ago there was some papers that showed that it impaired behavior, particularly olfactory related behavior in fishes and some invertebrates. Then she saw some contrasting papers recently. They're saying that that wasn't correct. So just wanted to see where that stands and if there's anything further that's been done on the issue.
Yeah. Hi, Judy. I haven't seen anything further from those papers that were trying. There was a lot of debate going back and forth between the different research teams that found one response and the other team found the other response. And, you know, this is the thing with ocean acidification. Well, not just with ocean acidification, but with science in general. You know, the idea with science is that, you know, you want to be able to recreate something someone else did. I mean, that's the whole scientific process, right? You do something and, you know, you put your methods out there, you put your papers out there, and you should be able to have somebody kind of reproduce that. And sometimes it doesn't work. And that's what happened in this case, where one research team, you know, did the experiments and found impacts to the olfactory senses in fishes. And then another team tried to do the same thing and they got different responses. And it's really important because, you know, the responses matter. So, and it can be anything from differences in the approach that they took to measure those. So it's really hard to say really what happened there. I'm not involved in that debate, but I am very interested to seeing some future work that continues to test those impacts. So we'll see. I don't have a good answer to that, except that I'm also waiting to see some more research to see, you know, what some other groups might find as well, to see if there is a consensus either way.
Yeah, we have another question from Wendy Walsh. She had a question about Donax, a small bivalve that lives on the beach in shallow ocean waters. She wanted to know how periods of low omega might impact the species and their role at the base of the food web.
So what species is it again? I don't think I'm familiar with it.
Donax.
It's a shellfish?
It's a small bivalve that lives on the beach and near shallow coastal ocean waters.
Okay.
So I think
Like
it's
in
like
the
in the
sand.
surf zone. Yes.
It lives, okay. Okay, so it probably has a foot that goes down into the sand. Yeah, like I said, I'm not familiar with the species. The good news about some of those species that live right at those shallow areas where there's a lot of fluctuations is that they're typically pretty tolerant of changes. They can tolerate a pretty wide range of things. I can't say that with 100% confidence for this species because I'm not familiar with it. But in general, species that live in these really kind of just dynamic areas where there's a lot of changes in the environment, in their environment, they typically are more tolerable of changes. The one thing to be, you know, to think about is that, and I didn't mention this in my presentation, but some studies are now kind of showing that with ongoing warming and ongoing acidification, that those swings will get larger. So you'll see more extremes. So you'll see extremes on the positive end and then extremes on the not so good end. So you'll see really low pH, really low dissolved oxygen, and then periods of really high pH and really high DO. And so that now there's a bunch of studies that have started to publish but are still ongoing, looking at the impacts of those swings on organism function. Like, because they might have to just completely shut down physiologically during those really extreme periods? And then how might that impact growth and reproduction and things like that? So those are some of the things we think about for organisms that live in those kind of dynamic areas.